Hi guys, and welcome back to the Fungi Files. Today, we're continuing our ambitious journey with Amanita Muscaria, the fairy tale mushroom. In this part of our experimental project, we'll dive deep into selecting host plants, the soil preparation, as well as integrating and ordering MHB, mycorrhizal helper bacteria, to encourage healthy mycorrhizal relationships. Let's take a look at the mycelium and then order some MHB so we can continue on to the next steps. Here is the Amanita Muscaria liquid culture, which is growing out very nice. And all three of our agar dishes have grown out fantastically as well. The mycelium for both of these is very nice, white, and clean. That's a good sign that this culture is healthy and well established. This mycelium will eventually inoculate our host plants, but before we get there, we have to prepare some other things first. The next step in our journey is to order the MHB itself. The mycorrhizal helper bacteria that we'll be looking at today, Pseudomonas fluorescens, will enhance the nutrient uptake in our plants, encouraging healthier root systems and setting the stage for a strong mycorrhizal relationship. Let's order that P fluorescens. On the computer, we can now head over to Carolina Biological Supply, search for Pseudomonas fluorescens, select our slant culture, check this beautiful slant out. We can order it, check out, and get it on the way. Once it arrives, we'll start culturing it out. While we wait though, we'll begin our nursery visit to select our host plants and prepare our soil mixture. Now, it's time to select the plants we'll use for this mycorrhizal experiment. I'm here at the nursery, and I've got a few plants in mind. With the insight of some helpful staff and my personal research, I've narrowed it down to a few hosts to start this. Now let's talk about the plants I selected for this first round of experiments and why. Now, at my local nursery, I could not find any ryegrass, so we started with some Suwannee blue-eyed grass, which is a singular grass structure. We'll be able to spread this out into separate grow containers and allow that to grow out. We have also started with a azalea. This is a Florida flame azalea. The reason I've chosen both of these is because of their high acidity content in the soil, as well as their ability to form fungal relationships. And we're hoping that in a controlled environment, we can use this to hopefully replicate the same environment indoors. And finally, we bought white clover as well, as this is another acidic balancing plant with good roots that we can hopefully form a microbial and fungal connection with. And the white clover introduces nitrogen to the soil, which is another factor to Amanita growth. We're using all of these because, in theory, they should be able to replicate the nutrients, the acidity levels, and hopefully the same environment in that soil. The hope is that with one of all of these, and the addition of MHB, we'll be able to create a stable soil environment. Check out that little turtle. Now, we're back from the nursery. We have our azalea, we have our blue-eyed grass, and we have some grow containers. For this beautiful azalea, we're first going to start by making some propagations using some rooting powder, some soil, and some smaller pots, so in a few years we can have future clones of this plant to continue the experiments with. We'll also be transferring this to a bigger pot, making sure to remove any dead roots and a lot of this old soil mixture so we can put it in a new composition of soil, and with the added space, encourage new root growth. The idea is that we can infect this new root growth with our MHB, let that culture, and eventually add our mycelium to it. Now, the azaleas do take quite a long time to establish, so that's why the first step of this is going to be making these propagations. Now, to do this propagation, we'll be taking off a heel cutting, which is just gently broken away at the woody part of the base, and we'll be inserting this with some rooting powder. This is not a necessary step, but does help speeding up the rooting process, and placing them each into small, well-draining soil pots and covering the bases lightly. Over the coming months, we can monitor the growth of these to make sure that the roots are starting to spread and sizing up the containers as it needs. This propagation will hopefully give us a continuous source of these azaleas to continue our Amanita experiments in the future. Let's go prepare our soil so we can transfer these propagations and transfer the azalea to a bigger pot. Now that we've selected our host plants, it's time to prepare our soil mix that'll serve as a foundation for this experiment. Amanita muscaria thrives in acidic, well-draining, forest-like soil. And today we're going to replicate that environment using a mix of materials. Let's break it down. We'll start with our base, which will be this forest soil C-mix. It's rich in organic matter and already contains peat moss, perlite, and sandy loam. Great for moisture retention and drainage. Next, we'll be adding in a little bit of Wisconsin sphagnum peat moss. This will further lower the pH level, ensuring that the soil is acidic enough for Amanita muscaria to thrive. We'll start by filling up our container about 75% of the way with this soil mixture. Now with the bucket about halfway full, we can add in about 20% sphagnum moss. I'm gonna add in about a third to a half of that bag. And to improve the soil structure and drainage, I'm gonna be adding a little bit of vermiculite, a little bit of perlite, and some pine chunks. This will make up about 10% of the mixture. And we'll add in a small amount of perlite and vermiculite. This will keep the soil light and airy, mimicking the natural forest floor. Now it's time to mix this up and get ready to propagate our plant. Now with this thoroughly mixed up, we're going to go ahead and fill these smaller propagation containers I'm going to be using. Now let's get our heel clippings off of our azalea. We're going to do this by taking some of these branches and gently breaking it away at the woody base, so you have fresh green. We're going to take our rooting powder and just thoroughly coat the bottom of that and plant her in. One azalea propagation complete. I'll pull the next one from farther up. Same ordeal. And lightly into that topsoil. And we're going to do one more. Nicely coated. And into that topsoil. These little baby bonsai azaleas can be monitored over the upcoming months with good watering, not overwatering though, and watch for root development. These can take a very long time, especially with this being a deciduous azalea. It's going to be a lot harder to root, which is why I took 3 of them. And if this plant continues to grow, we'll be able to make more. Now for the bigger part of the project, we have to transfer our azalea into this with our fresh substrate to get new root development so we can infect it with the Pseudomonas fluorescens over probably in about a week or 2. I'm going to go ahead and start by pouring a good portion of our mix in. I'm definitely going to have to make more. Trying to just knock this away so I can see what the current root structure looks like. With it being deciduous, it is going to be pretty hardy. And I could trim some of these roots. I don't think I'm going to do that yet. I just want to get it replanted for now so we can get some new root development going. Put the rest of that in there, and this may be good for now. Now, with our azalea repotted, ready to acclimate and develop new roots, we can go ahead and move on to our white clover and to repotting our blue-eyed grass. Now we can prepare the pots for our grass. As well as the containers for our white clover. We can now work on breaking up the blue-eyed grass. We're just trying to gently massage this apart. Oh. Finally, we have these separated. First one done. Number 2. And number 3. With our bluegrass complete, we can move on to introducing our white clover seeds. Oh, that may be way too much. I have no clue. We're going to sprinkle a nice amount and cover them in topsoil. At this point, we can go ahead and water everything gently with dechlorinated water. I'll show you in a future video how to make water, but we'll talk about that later. We'll wet these all down, let them grow out over this next week or so they can grow new roots or start to grow with this white clover. Then we can infect it with the MHB, and after we've given that about a week or so to get situated in the roots, we can go ahead and inoculate it with our Amanita Muscaria Mycelium. Now, we've moved everything into new containers. This allows for new root development. We want to do this before introducing the MHB, as we're looking to create ecological balance instead of being completely sterilized. I think that we can achieve ecological balance by using this P. fluorescens as well as organic nutrients. And it's here. We got our P. fluorescens bacteria slant from Carolina Biological. We're not going to open this quite yet. In a future video, I'll be learning how to work with bacteria, with transferring from slants to agar, as well as making a liquid culture to grow this bacteria culture out for use in our plants. After about a week or so of the new growth of the plants, we'll introduce a diluted solution with this bacteria culture and apply it to the root system. This inoculation will protect the plants and support a healthy mycorrhizal relationship. And once the MHB has finally established itself in the root zones, we can introduce the Amanita Muscaria liquid culture. Hopefully, after this first round, we'll see some success in achieving indoor fruiting. However, I expect there to be many more iterations of this before we achieve actual fruiting success. Moving forward, I'll be monitoring these plants to see how they interact with the Amanita Muscaria Mycelium. As I fine-tune this setup, I'm probably going to be looking at other species of plants, maybe legumes, legumes,
Legume.
as they also boost nitrogen levels. And in future videos, I'd like to attempt selective breeding to see if we can work on developing an Amanita strain that can be more successfully cultivated indoors. Thanks for following along on today's experimental journey. If you enjoyed today's content and want updates on this unique project, be sure to subscribe, like, and hit that bell button for notifications. If you have any suggestions or questions, be sure to leave them down below. Until next time, keep growing and keep exploring.